Hello everyone, and welcome to Biopedia. Today's episode is, like we've done a few times now, an adaptation of something I've written previously to the podcasting format. In this case, it's an article I wrote in a bid to be published in an undergraduate science journal in my second year of undergraduate studies. So, I hope you enjoy. Cultures throughout history seem to have loved tracing themselves back to a founding couple. The Greeks had Deucalion and Pyrrha, the only humans left after Zeus caused a flood to destroy the rest of the world. In Norse mythology, Ask and Embla were created by the gods from the trunks of two trees. The Bible, meanwhile, famously has the story of Adam and Eve, whose fall cast humanity out of Eden. In the modern world, these stories are viewed as allegory, metaphor, or simply origin stories. However, Adam and Eve still survive in one major field: genetics. Although the story of two founding humans is just that, a story, the tale survives when discussing the DNA that can only be inherited from either your mother or your father, the mitochondrial DNA and the Y chromosome. Let's start with Eve's side of the story. Mitochondria, compartments within the eukaryotic cell used to generate the energy currency ATP, were discovered to have DNA of their own back in 1963. A letter to Nature from 2000 specified that mitochondrial DNA evolves at a fairly constant rate. In fact, the evolutionary rate of mitochondrial DNA within chimps and within humans is not too dissimilar, with the exception of a section of DNA called the D loop. This general constancy makes mtDNA, which is the shorthand version of mitochondrial DNA, a useful tool for assigning a date to evolutionary events. The geneticist and author Brian Sykes first moved towards mitochondrial DNA in the 1980s, after deciding to try and extract DNA from old bone due to the fact that mitochondrial DNA outnumbers any other gene in the cell by about 100 to 1, another useful property of the mitochondrion. The mitochondrion is, with a few exceptions, only ever inherited from the mother, as the sperm cell's mitochondria do not enter the egg cell with it. This means that a line can be traced from mother to daughter continuously, forming something like a matriline. In theory, this means that there should be one person from whom every human alive today inherited their mitochondrial DNA, mitochondrial Eve. When this so-called mitochondrial Eve lived has been the subject of debate across the decades. The 11th edition of the Penguin Diction of Biology, published in 2014, gives an approximate date range from 400,000 to 600,000 years ago. By contrast, a 2019 estimate placed the origin of one of the oldest subsequent branches on the mitochondrial family tree between 240 and 165,000 years ago with 95% confidence, based on studies of this basal so-called L0 branch. Interestingly, this seems to fit with original studies back from 1987, which analysed 147 different people and concluded that MT Eve probably lived about 200,000 years ago in Africa. In the thousands of years since this individual lived, the mitochondrial family tree has split into many different groups or haplotypes, something covered extensively by Brian Sykes in his 2001 book, The Seven Daughters of Eve. We'll get into this more at some other episode, I'm sure, but as the name suggests, Sykes created 7 daughters of this mitochondrial DNA ancestress, meant to represent the different haplotypes on the tree. The so-called clan mothers are given the names of Ursula, Xenia, Helena, Velda, Tara, Katrine, and Jasmine. These clan mothers were given names and identities by Sykes to allow people to connect with the stories of these individuals. An example he cites in his book relates to the discovery of a maternal line relative of Ötzi the Iceman, who died around 5,000 years ago and was preserved in the ice until his discovery in the Italian Alps in 1991. This relative they discovered promptly began to think of the Iceman more as an individual rather than a news story. This is something Sykes aimed to recreate when discussing these mitochondrial ancestresses. Moving over to the patriline side of things, Y-chromosome Adam is pretty much the male equivalent of mitochondrial Eve. The Y chromosome, passed from father to son, contains the SRY gene, also known as the testis-determining factor. Just as in the case of mitochondrial Eve, Y-chromosome Adam is the hypothetical individual from whom every father-son line descends, and thus whom every male on Earth has ultimately inherited their Y-chromosome from. Once again, though, we find that people have had differing opinions on when this figure might have lived. According to an article in Nature News, original studies placed him 100,000 years ago, millennia removed from mitochondrial Eve. However, a population geneticist by the name of Carlos Bustamante believes that this picture might be skewed. The calculation of when any common ancestor like this lived depends on the number of breeding adults in a population. If polygamy were practiced at any point, the number of males that would have been able to successfully pass on their Y chromosomes would be smaller, thereby obviously altering the results. You've got a different number of individuals near the top of the tree. Two studies, one by Bustamante and one led by an Italian called Paolo Francalacci, both independently concluded that these two figures, mitochondrial Eve and Y-chromosomal Adam, lived at the same time. However, they disagreed on the dates. Bustamante's estimates for these two intersect between around 120,000 and 148,000 years ago. By contrast, Francalacci pushed Adam's existence back to about 180 to 200,000 years ago, roughly the original 1987 estimates of Eve's age. For his part, Bustamante suggests that the discrepancy in the two figures may be to do with populations they studied and how their molecular clocks were subsequently constructed. After all, if you estimate a different mutation rate for either the Y chromosome or the mitochondrial DNA, the ticking speed of the so-called clock would be different, thereby altering the total number you end up with. The Y chromosome and mitochondrial DNA are also useful when considering broader human history and prehistory. For example, one study described how both most basal, i.e. ancient, branches on the Y chromosome genetic tree are found in predominantly African populations today, confirming the African origin of humanity. Furthermore, the Y chromosome has been shown to become progressively less diverse the further away you go from the Eastern African origin point of humanity, as discussed by a paper by Chiaroni and colleagues published in the Proceedings of the National Academy of Sciences in 2009. This correlates with the idea that humanity expanded out of Africa only once, with these populations going on to found new colonization efforts. This gradual wave outwards stepwise is something called the serial founder effect. So you've got Group A founding populations which are Group B in a ring around it, and then Group B founds Group C in a ring around them, and so on. In effect, these small fragments of DNA, the mitochondrial DNA and the Y chromosome, can help us to rediscover our story in a way we otherwise perhaps could not. The literature is still divided on the subject of the two figures we're discussing today, as all the various dates mentioned above have demonstrated. It's unlikely to be settled anytime soon. Now, obviously these studies are not connected to the biblical stories in any sense. Neither are these papers indicating that these two figures even met. As we've seen, the dating of these two ancestral figures can vary wildly from decade to decade. The names Adam and Eve themselves are just that, names from a different context which society has retained as useful constructs when discussing genetic terms. However, it's still interesting to speculate that genetic origin points we've artificially labelled as Adam and Eve may have lived at the same time, just as the stories they come from would have told us. That brings us to the end of today's episode. As always, thank you all for listening. Feel free to get in touch at the show's email address for any questions or comments. Until next time, have a great week, everyone.